Good afternoon. Thanks, organizer committee provide us the opportunity to share the work. I'm Zeng Li from National Neuroscience Institute of Singapore. Today, I would like to share our recent discovery using the patient iPS-derived cerebral organoids and mouse model for neurological disease study. Along with technology advancement, whole exome sequencing, GWAS, and multi-center large cohort screening, people found neurological disease no longer sporadic. They are linked with the genetic mutation. So working with Dr. Adeline Ng and Professor Tan-Yin King from NNI, they have identified various genetic mutations from neurological disorders. And for instance, this NOTCH3 mutant linked to the vascular dementia, and PAS1 mutant linked to the Alzheimer's disease, and NOTCH2 repeat expansion linked to the NIID disease, as well as the CSF1R linked to LSPs. However, due to the lack of the proper modeling, the functional role of these genes are not fully elucidated. Particularly, how does these mutations contribute to neurological disorders remains unknown. So today's presentation, I will focus on the finding from the NOTCH3 and NOTCH2 works. So first, I would like to share our using the patient iPS-derived cell types to investigate the effect of the NOTCH2 Asian with genetic mutants on the blood-brain barrier. And second, I would like to focus on investigating the NOTCH2 NLC GGC repeat expansion in neurodegeneration using the mouse. So as we know, dementia is an umbrella term for loss of the memory, that including the Alzheimer's disease, vascular dementia, and Lewy body dementias. In Singapore's Alzheimer's disease and vascular dementias account for the majority of the dementias for both young and late onset of the dementia. So vascular disease is a small vessel disease, subcortical lesions, and with small, the red matter lesions, and also caused by other disease such as the stroke, hypertension, and the diabetes. So CADSO is a genetic form of the vascular dementia. The symptoms occurs 20 years before the onset of the dementia. And this out of the 3 CADSO cases will develop the vascular dementia. So this is better to monitor the disease progression and prevent the disease deteriorate. So here is the clinical feature of these CADSO patients. The MRI imaging show this red arrow indicate the Lacan infect, which lead to the ischemic events. And the orange color indicate the red matter lesions, the axon degenerations. So apart from being the small vessel disease and red matter lesion, CADSO skin biopsy reveals the presence of the granular osmophilic materials in the blood vessel walls. So these GOMs are intranuclear accumulation composed of these NOTCH3 proteins and other nuclear aggregations that contribute to the damage and dysfunction in the CADSOs. Additionally, this EM study has shown the abnormality in the smooth muscle cells and the extracellular matrix of blood vessels, which can impair the blood flow and contribute to the ischemic injury. So this NOTCH3 gene is associated with the CADSO patients. NOTCH3 is a transmembrane receptor protein. At the N-terminal, the ectodermal domain consists of these 34 EGFR, and each EGFR contains large members of the cysteine residues. And most of the NOTCH3 mutations have the odd numbers of the cysteine residues in the EGFR domains, which can interfere with the proper formations of the disulfide bonds between these residues, and leading to the misfolding and accumulation of this NOTCH3 ectodermal domain in the cells. There are about 270 mutations in the NOTCH3, and this has been linked to the CADSOs. And this R544C, is the most common mutations reported by the Asian patients in China, Korea, and Singapore. Through the large-scale genomic study in Singapore population screening, it reveals that almost 1% of the Chinese population carries the specific NOTCH3 risk variant R544C. However, due to the lack of disease modeling, the function and regulation, as well as the disease pathogenesis, remains unknown. So because Notch3 mutant linked to the vascular defects, we speculate it is important to regulate the BBB and leading to the BBB dysfunctions. So our objective is to develop this in vitro BBB model to recapitulate the disease phenotypes and to be used as the drug screening platforms. To elucidate the human models, establish the phenotypes, we first reprogram the IPS from the PBMCs. And then the PBMC will then reprogram to IPS, and to reduce the heterogeneity between the samples, we correct the mutations using the CRISPR-Cas9, which serve as the anthogenic controls, and subsequently we characterize the IPS. So next, we differentiate the BBB cell types from the IPS will carry out. The endothelial cells will differentiate from the hematopoietic progenitor cells and mesoderm cells, and then sorted for the CD144 positive cells. And parasites will also differentiate with the similar protocols at the endothelial cells and sorted for the non-CD144 positive cells. The astrocytes was obtained from the day 19 cerebral organoids and disassociated into the single cells and sorted for the glass positive cells. So this differentiated cell type was stained with the cell type specific marker C31 for the endothelial cells and the PDGFRB for the parasites and GF80 for the astrocytes. So to assess the endothelial cells vascular defects, we first performed the angiogenesis analysis. We found reduced branchings in the Notch3 mutant cells, and subsequently we conduct the migration assay using the scratch bone assay, and we found that these mutant endothelial cells takes longer times, shows the delay in healing, suggests the impaired proliferation and migration. In addition, we conduct the EER assay. It shows the tightness of the barriers is low. This indicates that Notch3 mutants may have vascular dysfunction seen in the CAD cells. So next, we conduct the bulk RNA-seq works to identify the pathway altered in these mutant endothelial cells, and we found they were involved in the branchings and vasculature development. And the altered pathways in the mutant endothelial cell are involved in the ECM organization and vascularization regulation. And this dysregulation pathway altered really appears to be consistent with our functional analysis. So to elucidate the vascular network defects, we first generate these 3D BBB-like macrotissues, and we utilized this 3D tissue culture platform called the organic plate. The plate is in a 24-well format that utilizes the principle of the microfluidic chips, that it consists of microfluidic chips, but instead of individual lengths that specific for the specific cell, the plate has more heterogeneous chamber and allows for larger samples like spheral and multiple cell types to be seeded. So this platform allows us to conduct the immunostaining experiment directly on the plate and easy access to the sample for downstream protein assay and the gene analysis using the plunger. So what we do here is that we see these iPS-derived endothelial cells, parasites, and astrocytes, and we have mixed with the hydrogel into the chamber. So after 7 days in cultures, we characterize this BBB-like microtissue formation. So to characterize the BBB-like microtissue formation, we first conduct the immunofluorescence BBB-like tissues in various cell type markers. We show this visible lumen in the X and YZ profile with the positive staining for various cell types of endothelial cells, smooth muscle cells, and parasites staining. And our model shows the vascular-like and lumen-like morphologies. Next, to define the hyperperfusible ability of BBB, permeabilization assays with various sizes of the fluorescent labeled dextran was conducted, and we found that there's no thickness of the dextrans, even with the smaller size of the dextrans, hence demonstrating the tightness of the BBB. So these permeabilization assays show the vascular networks formation appear to be perfusible and display the barrier integrity. So next, we incorporate the Notch3 mutant into these platforms to elucidate if there's any vascular networks phenotypes. And we found the vascular formation is sheet-like, does not appear to form the distinct networks in the mutants. So in addition, this mutant macrotissue showed increased permeability is indicate the loosed barrier. So this platform serves as a promising tool to study the vascular formations and structure as well as the protein drug screening platform. So to summarize, what we show here is the mutant Notch3 have the decreased migrations, angiogenesis, and barrier tightness. And these mutant BBB-like macrotissue show the defect vascular networks. And leveraging on this neurovascular-like unit, we aim to develop the novel therapeutic drug treatment in the cardiac patients. Now I would like to switch gear to talk about the other model system study using the mouse model to investigate the Notch2 N2C GGC repeat expansion in neurodegeneration. So as we know, the neurodegenerative disease characterized by the progression of the loss of neuron in the nervous systems, and it's leading to the impairment in memory, cognition, behavior, sensory, and the motor dysfunctions. So a set of the interconnected pathological hallmarks is commonly shared across the neurodegeneration is the protein aggregations. Here are some examples of the neuropathological lesions showing the abnormal deposit of the disease-specific protein. For instance, Alzheimer's disease is A-beta and tau, and Parkinson's disease is alpha-synuclein. Huntington disease is the Huntington protein, and in the ALS is the TDP and TDP43. So understand the mechanism of the protein aggregations provide the common framework for dissecting the neurodegenerative disease pathogenesis and identifying the therapeutic target. So the disease we are focusing on is the typical example of an aggregation-related neurodegenerative disorder called the NIID. It's a neuronal intranuclear inclusion disease. This concept of the NIID was originally described in an autopsy patient who was found to have widespread intranuclear inclusion throughout the nervous system and other organs. Since year 2000, an increased number of NIID cases has been identified, especially in the East Asia. So in clinicals, the typical MRI feature is the hyperintensity along the cortical medullary junctions on DWI imaging suggests the acute toxicity injury. In addition, the white matter lesions observed in the subcortical regions and the cerebellum, indicating the widespread myelin abnormality and the axon degenerations. So pathological examination, especially through the skin biopsy, reveals that the protein, the enrichment intranuclear inclusion, they are positive for the ubiquitination marker and P62 and the SUMO1. So recently, the long-read sequencing has been identified, the GGC repeat expansion in the Notch2 and LC genes, a human-specific parallel of the Notch2 involved in the brain development as the causative mutation of the NIID, with particularly high prevalence in Chinese, Japanese populations. So typically, the GGC repeat expansion below 40 repeat considered normal, and 40 to 60s are intermediate, who will show the mild symptoms, and above 60 will consider the pathogenic. How does this GGC repeat expansion mutation lead to the pathogenic phenotype observed in the NIID remains unknown. The mechanics study has revealed that the GGC repeat expansion is located in the 5' UTR region of the N2C genes. So this repeat expansion indicates the translation from the upstream open reading frame and to the production of the novel small protein called the N2C polygenes. So this N2C polyg consists an extended stretch of the glycine residues, and this polyg protein is prone to nuclear aggregations and forming the intranuclear inclusion, which in turn contribute to the white matter degenerations. So there are multiple model systems, including the Drosophila model, mouse model, and the human models, has been developed to investigate this downstream effect of GGC repeat expansion in the N2 NLCs. In these model systems, several common biological processes are consistently disrupted, including the mitochondrial dysfunctions and the nuclear cytoplasmic transport and the ribosomal biogenesis and myelinations. So in parallel with these model system studies, a multiple clinical cohort study has reported associated with the GGC repeat expansions. In year 2020, our collaborator, Professor Tang King and Dr. Edwin Owens from NNI were first to screen for the NOTCH2 NLC GGC repeat expansion in Singapore PD cohort. So out of these 1,000 sporadic Parkinson's disease patients, 13 of them has been identified carrying the GGC repeat expansions. And among these 13 GGC repeat expansions, about 40% show the serine AGC insertions in the repeated sequencing, which was associated with more severe clinical phenotype. So to elucidate the pathogenesis in association with Parkinson's disease, we generate the mouse models carry the intermediate repeat with and without serine insertions. This work was conducted by postdoc Haital and Zhi. So first, we investigate if there is protein aggregation formations. Immunofluorescence staining of the N2Cs showed that there is a fiber-like protein aggregation in mouse cortex. Next, we determine whether there is a neurodegeneration in Parkinson's disease-associated protein expression in these NOTCH N2C serine insertion mouse. The Western blot and immunofluorescence shows these N2C serine insertions induce the hyperphosphorylation of alpha-synuclein in both soluble and insoluble fractions of the mice midbrain regions. So apart from the midbrain regions, we also investigate in the mouse striatoms, which is another important brain regions for Parkinson's disease. And we found this N2C serine insertion induce the hyperphosphorylated alpha-synuclein and promote the DA neuron loss in the TH. So next, we conduct motor behavior analysis. This rotor rod, cylinder test, pole test, as well as the open field test have shown that this N2C serine insertion mice display the motor behavior deficit. So motor behavior is associated with the neural excitability. To assess any abnormal neural excitability in mice, we conduct electrophysiology recording of action potential. And we found there is an increased action potential in the prefrontal cortex neuron of the N2C serine insertion mice without the serine insertion mice. This demonstrates the hyperexcitability in mice. So at white matter lesion is associated with dysregulated myelinations. To detect the myelination status in the N2C serine insertion mouse cortex, we conduct the transmission electron microscopy to direct visualize the myelination neurons. So after the coronal sections, the 1 milliliter cortex near the corpus callosum, as shown in this red box, which is the motor neuron region, was cut for resin embedding. So by comparing the myelinated axon, we found that there were more myelinated axon in N2C insertion mice, which suggest the hypermyelinations in mouse cortex. So to summarize, we have shown that N2C mice with serine insertion show the Parkinson-like phenotypes, and also there is a hyperexcitability and the motor deficit, as well as a hypermyelination in these mice. So given that recently in clinical, we have found that mild intermediate repeat expansion of NOTCH2 NLC are associated with Parkinson's disease. Here we are the first times we identified the new pathological link in the mouse models expressing the intermediate repeat with serial insertion. Our findings can lead to new therapeutic based on the potential target we identify. Last but not the least, I would like to thank our collaborators I have highlighted during our presentation, as well as the funding agent. Thank you for your attention.